agreed to have this session recorded as well, and then I'll turn off the recording at the Q&A portion so we can be frank with our questions. And also, he has agreed to send out the slides after his presentation. So Dr. Sean Mooney is the Chief Research Information Officer at UW-Medicine, the Director of Informatics at the Institute of Translational Health Sciences, an Associate Director at the National Alzheimer's Coordinating Center, and a Professor in the Department of Biomedical Informatics and Medical Education at the University of Washington. As Chief Research Information Officer, he leads a growing team that contributes to a number of national research networks and activities, such as the National Kidney Precision Medicine Project, the National Alzheimer's Coordinating Center, and the UW-Clare Center. His team continues to develop an EPIC EHR research team for supporting research in the EHR at UW-Medicine. He is leading the newly founded UW Institute for Medical Data Science. His research interests focus on data science applications in biomedicine, particularly in understanding the underlying molecular causes and inherited genetic diseases and cancer. The researcher perspective is important to every aspect of data sharing, from planning data sharing to moving through the research activities and on to the sharing and reuse, which is what Dr. Mooney will be sharing with us today. Dr. Mooney, you can go ahead and begin your presentation now.
Awesome. Can you hear me okay?
Yes.
Excellent. All right. It's super pleasure to be here. Thank you for the really kind introduction, and it's wonderful to meet all of you. I'm hoping you'll enjoy this presentation. This is going to be a little different than my normal talks that I give. One, I'll limit this to, maybe 30 minutes or just a little bit over, because I want to leave plenty of time in the hour to have questions and answer and Q&A at the end of the presentation. And it's also going to be a little different in that this is a follow-up from a talk that I gave that was relatively short at an NIH-hosted meeting on generalist repositories that happened in January, I think. And so what I'm going to do is give some of the slides, or maybe all the slides I gave in that talk, and then I'll talk, since I have a little bit more time, I'll talk about some of the work that we're doing to develop what I would call cyber infrastructure for national data coordination for biomedical data to make data available. You've heard about two of them in my intro, the Kidney Precision Medicine Project and the National Alzheimer's Coordinating Center. And I'll briefly talk a little bit about what we're doing at UW-Medicine, the health system. So it's a super pleasure to be here. I also want to, just make a little bit of a comment that I'm really pleased to see both librarians and informatics and biostatistics and population health and public health kind of all here together. I think that these are groups that should collaborate well together at institutions and don't always collaborate well, sometimes can be siloed. And we've had a long-standing collaboration with our health sciences library that's led by Tanya Barden here at the University of Washington School of Medicine. And that's been really important and impactful for supporting kind of the informatics platforms that we roll out to support biomedical research. So I'm really pleased to see that. So what I'm going to start with is just kind of an overline intro into data sharing and how we see data sharing. My team has been in the trenches working with professors and investigators for almost 30 years now. And I think we really have a good understanding of how data is generated, how it's shared, and how the world is changing with respect to data collection based on, technologies like the internet and mandates that come down from, like, say, the federal government that talk about a strategy that's more collaborative, more integrated, the concept of shared computer infrastructure across the nation and even the world for supporting for supporting, data generation, curation, and making data available for investigation, with the idea that we might be able to make better discoveries, we may be able to implement new methods better and improve human health and healthcare. So we all know, let's think about the investigator. We all know investigators, scientists are generating data at a crazy rate. And there's, I think, a consensus among our community that data should be shared outside of an individual publication. Publication supplementary materials often have data in it, and it's often a table or a summary, and is really insufficient to reproduce the work or to extend the work further. But the sharing of data is really most impactful when when we make some effort to make it useful within an ecosystem of other data sets. And that is like, how can we make data integratable with, like, say, a paper by a competitor, and enable analysis across either both data sets or a comparison of those data sets. And immediately when we think about making data useful within an ecosystem, we think about how we might standardize that data to be harmonized or harmonizable with other data sets that have concepts within them that are the same or similar. And I think today, if you were to go into the labs in your university or to professors or investigators that you work with, I think there's probably a substantial gap in fluency, but how to actually share data in practice and what work needs to go in to make that data useful. So let's talk about kind of the high level, 50,000 foot level view of researcher generating and sharing data. Today, that really involves, a researcher generating a data set. They write a paper. We may be required to share that data set with a collection of data in some sort of a central repository. I'm thinking specifically outside of the publication. There are a number of national repositories that are specific to certain domains. And then we're also seeing the rise of a concept that I'm hoping people walk away with, the idea of a generalist repository, that is a repository that can share data that isn't built to support any specific disease or scientific area, but really with the purpose of sharing data for research and for reuse of that data. And then data scientists can collect data from this central repository and do new science. There's lots of examples of how this has been really successful and is a useful model. Sustainability is very, very difficult of these collections of data. Something worth mentioning, the federal government has struggled, I think, to support infrastructure and are still continuing to make efforts in that area. But I think that that's still an open, an open challenge for the future. Is if we build a database, how do we measure its value and impact? How do we measure whether the dollars that are being spent are, really showing the value and the publications and the scientific advancements that we expected from the beginning? How do we create new collections of data as new technologies happen? How do we sunset ones as new technologies go away? I should mention that it's important to note that data sharing is important here, and data sharing actually happens twice. Data sharing happens when the data set is contributed to the collection of data, and then the hosts of that collection of data then share data to those scientists. And I think that's important from the perspective of a data curator or the investigator or the institution to really understand that that there may be multiple times data is shared between when an investigator generates the data and when a scientist external to that investigator uses it. So researchers contribute to data repositories. There are many examples of these. I'll talk in detail about two of them here in a second. Investigators often have, both pressure to share the data and to make that data useful to others. And one of the primary drivers in biomedicine of that is the National Institutes of Health. They have a strategic plan. You can download it. It's a PDF file. You can look through and see how much, information and informatics and data sharing and data science all pop up throughout that strategic plan in many, many different places. And there's also an NIH strategic plan for data science specifically that was developed out of the Office of the Director that, you know, really speaks to a more integrated and harmonized nation of biomedical researchers that are generating data. So instead of having individual papers with, you know, supplementary information that is data, and instead of having siloed data repositories, the future state may be really truly harmonized resources that are integratable across each other, and, you know, could be you know, considered cyber infrastructure that is built out of, like, say, a cloud-like environment. Maybe we can talk more about that during the questions. But in order to get there, we must think about all the important things that we need to think about to make data harmonized. And it starts at the beginning in study design, just like we do biostatistical study design when we determine how many patients we need to do a study or how many data points we need to be able to have the power to detect or you know, to detect or to you know, accept or reject our hypothesis. We also have to start and think about informatics and data at the beginning. And this is very underserved nationally, even today. And what do I mean? Well, so if we collect data, we need to make sure that we're collecting data in a common way. Like, we need to have common data elements that can be shared amongst other investigators. If we're doing clinical research, for example, and we're collecting educational history, just as an example, a common data element would be, how do we collect educational history such that we're all collecting educational history in the same way? Like, some may say elementary school, middle school, high school, you know, college, graduate school. Others might say first grade, second grade, third grade, fourth grade, fifth grade, et cetera. That's not necessarily harmonizable or common, and we want to be able to agree on common standards as much as possible. That also includes nomenclature. Like, how do we when we you know, do annotations or collect data, we want to make sure that the terminology we use are standardized. We use biomedical ontologies to accomplish this task. There are many examples of them. I would argue that the most widely used biomedical ontology in the world, if I can call it an ontology, is the International Classification of Diseases, or ICD-9 and 10, which are how we do diagnosis codes for the purposes of billing and you know, maintenance and electronic health record systems. But they're also in biomedical research, in biology, ontologies such as the gene ontology, which you know, which is an approach for annotating genes and proteins with their products, function, you know, biological processes that they're involved in, and where they end up in a cell after they're produced. There are also standard instruments for clinical research. That's basically how we put all of the common data elements together. And if we wanted to collect medical history or social determinants of health or demographics, we would want to have a standard way of collecting that data. And it's basically an assemblage of common data elements for doing that. And then finally, we also want to have standard approaches for metadata. And I think this is really hard. I think we haven't done a good job as a community for developing metadata standards. And I think it's something that we all should think maybe more carefully about as we move forward. So, for example, if we have an investigator that's got some cell line that they're doing you know, single cell RNA-seq with, and they generate a data set, which is basically the gene expression from transcripts that are sequenced within those cells, they would want to distribute that data set with standardized metadata. You know, what was the instrument and technology that sequenced? What is the cell type? What was the experiment? What's the publication? Who did it? What institution did it? You know, all those, what are the conditions that were done in? All of those sorts of things need to be there, but you also want them there and standardized so that if everybody's, you know, single cell sequencing data sets were made available, you want to be able to compute with them. And in order to compute with them, we have to be able to compute with the metadata. So FAIR, let's talk about FAIR. FAIR is a you know, very hot term. It's really taken off. It was from a publication that's been very highly cited a few years ago, and is really a basic framework for how we can do data sharing. and is an acronym for findable, accessible, interoperable, and reusable. You're probably familiar with that, but the idea is that for data to be open and available, that these all have to be addressed and true. And some of those things are really easy. Adopting standard file formats, submitting data to a repository, like that sort of thing is relatively straightforward to do, and many investigators do it. Some things are a lot harder, like the use of biomedical ontologies or those common data elements, the standardized instruments that I mentioned before, because you have to start and do that up front. And investigators in the clinical research world rarely agree on how to collect, on what those instruments should be to use. So it's very easy to get off track and not do that. And then some of those things are very, very, very hard, in my view. So for example, generating synthetic data that maybe if the data has protected health information in it, or sharing protected data under a data use agreement to another institution. I should also mention, sometimes the way we collect data is not standardized, and investigators are really passionate about the way they collect their data, and getting them to agree to a standard way to collect data is sometimes almost impossible. So let's say we wanted to, and a good example of this was how we used to collect bone mineral density for clinical research. There are a lot of ways to generate, to determine what bone mineral density is. And historically, that's been very non-standardized. Like some people could use an X-ray from like a hip, or some people use X-ray from a wrist. And at the end of the day, those data isn't really that analyzable or integratable, and you want to have a standard way of doing it, like saying we're only going to collect bone mineral density from like an X-ray of your hip or something. I don't know what the standard is now, to be honest with you, but you got to get a bunch of people to agree on that, and they may have a database of data they've generated that they're very proud of that was using this other way, and so they don't really want to change. It's a hard problem. All right, so there remains a significant knowledge gap, I think, as I mentioned before, in terms of enabling data to be fair. It costs money. It costs an understanding of information and informatics. It's sometimes seen by the investigators as being contrary to the analysis of the data and the discovery that's going to happen. Some investigators don't see value in other people using their data. So there are a lot of challenges up with that. Investigators, in my experience, almost always lean towards spending money up front on data generation, not necessarily the data or the analysis, the preparation of that data or the analysis of that data. It's always been a challenge. Like I work with geneticists and they want to do, collect patients and do genome sequencing. The first question is, well, how much money do we have? And then the next question is, well, how many genomes can we sequence with that money? With no, respect for any of the other things that need to be done to make that happen. They just want to know how many genomes I can get. And that's a problem. So, I think, as I mentioned, that investigators really should make informed decisions beforehand. And we should do that study design for informatics that I mentioned that is really a standard practice for biostatistics. And the data sharing requirements are not always strongly enforced, except in specific consortia with explicit requirements where often money is involved. Like the NIH says, if you don't share your data in this way, we're not going to fund you next year. And then immediately the data gets shared. The data collections. So data collections come in many, many forms. There are coordinating centers for specific funded consortia. That is, the NIH will say, we want to solve the problem of colon cancer. So what they might do is fund a colon cancer consortium. They'll fund a bunch of clinical sites that will recruit patients with colon cancer. They're going to test some specific thing. They're all going to do it in the same way. And then they'll also fund a coordinating center to manage administration, maybe specimen processing, and data management. And those coordinating centers collect data and then make it available. There may be curated knowledge bases. These are people that are funded to review papers, find the findings from those papers, and then curate them and make them available. Examples of that might be like genome databases or the protein databases like at UniProt or at NCBI, RefSeq, things like that. And then finally, there are open repositories that are aimed at data sharing that can be generalist, like Zenodo, for example, or other tools that enable, making data openly available. And these so-called integrated data collections, I can't emphasize enough, really require previous work to be done before submitting. The investigators must be resourced for doing curation of that data, and they must have open standards available at the time of that curation, or those standards won't be baked into the data. And to basically curate unstandardized data into standardized data has a lot of cost. Data sharing, that second step of data sharing from the repository to the individual scientist, is also challenging. You may have laws that you have to abide by. So, for example, protected health information or institutional policy could require HIPAA protection for data. There are privacy laws, such as the EU's GDPR, which when involved with human subjects data, have significant constraints on how research can be done. There may be requirements of funding for cyber infrastructure and security controls in your computer systems, like FISMA, which can be very expensive, very hard, and there's very little fluency for that. And it may be required to pull that out of your direct costs for your grant to support it. You have to have a data use agreement, very likely. Many investigators still don't get one. The institution should be prepared to build them. Sometimes that's not true. And what those terms might be, sometimes other institutions won't accept those terms, which turns into a negotiation. And sometimes those negotiations don't work. And therefore, the research doesn't move forward and the investigators can't share. And then finally, as we build databases, the terms of informed consent and the IRB can be very important and critical in clinical research for how that data is used. So for example, if we do a study at the University of New Mexico and a study at the University of Washington, and at the University of Washington, we consent patients to use their medical records and not at UNM, then we can't necessarily integrate that data or collect patient medical records as part of that study. And so we have to know that. And if we have 1,000 studies all collected together and they all have different consents, we're going to have to reconcile that. And that can be very expensive, hard, challenging intellectually, and make the research very, very difficult. And a lot of people use the cloud and in collections of data now. And pulling data out of the cloud has costs, and sometimes those costs aren't accounted for, and we need to really think about those resources, et cetera, when we do that. I mentioned before, we need data use agreements. So when an investigator shares with a collection, there's data sharing that happens there. There is a data use agreement that happens when the collection receives that data, but then there's also a data use agreement when scientists receive the data from the collection. And we want institutions to make this as seamless and easy as possible. But it's very important, because sometimes if we don't have this in place and the investigators aren't aware of what they're obligated to do, sometimes we can have bad problems in the future when we discover, things that may have happened outside of outside of what was agreed upon between the institutions around data sharing or data use. And then we often talk about things like persistent identifiers, like DOIs, and the NIH will often require us to have DOIs on data sets. It's possible. If investigators generally don't understand them, they don't understand why they're useful, and it's not totally clear that they're generated in a useful way because of that lack of fluency, not because they're not useful. All right. Cloud computing in academic medical centers. I'm going to talk a little bit about this in some of my examples. There's a ton of interest in the cloud as a facilitator of fair data. And that's because when we think of this concept of cyber infrastructure, we are thinking of this shared computing platform around data and technology and analysis of data. And that shared, kind of this national shared platform, makes the most sense in the cloud. And so understanding the cloud, understanding how to use it, understanding what it means to, develop across the cloud, all those things become important. And there's still this drive to take us to things like the cloud, but also concern about how much the cloud costs, how secure the cloud is, and whether the environments that we're building are really aligned with each other so that you will be able to achieve the goals that we want to achieve around fair use of data. All right. I'm going to transition a little bit here to talk about some individual projects that we think are enabling fair use of data and are contributing to the cyber infrastructure of our biomedical research platforms. And I'm trained as a chemist, and I did bioinformatics as an assistant professor, and now I work for a health system doing clinical research and clinical research informatics. And so I've kind of seen a broad spectrum of computational data collection and use. I'm going to largely talk about clinical research now because that's like what I think about mostly. And I'll do this for about 5 minutes, and then I'll wrap up at about 9:40, if that's all right, or whatever time it is there. I think when we think about open science, a lot of people brag about being part of open science, but I want to, just when I think about what those facets are, I think about biomedical ontologies and these standardized data models and terminologies. I think about these open and fair data sets. I think about these open and standardized common data elements and common data instruments. I think of open software. I think of open science, like what we call community challenges, where data scientists or analysts can work on a single data set with having many doing the same experiment and then comparing their results to each other. And then I think about, opening and standardizing machine learning models or advanced statistical models such that we can move them around and apply them to data that they weren't necessarily built initially or published using. So let's talk about 2 examples, and then I'll wrap up. We're the technology backbone with the University of Michigan for the National Kidney Precision Medicine Project and the National Kidney Atlas Project, as well as the National Alzheimer's Coordinating Center, both of which collect our national collection of data around clinical research and Alzheimer's and kidney disease like chronic kidney injury and acute kidney injury. Chronic kidney disease and acute kidney injury. KPMP's been around for about 5 years. It's funded by NIDDK. Behind everything is a technical platform that collects a wide range of different data sets. And we have a team that really supports, collecting the data from the investigators in a standardized way, curating that data, and then making appropriate data available. It contains a lot of clinical data, including EHR data and about 10,000 clinical data elements that are collected by research coordinators, all of which could potentially contain PHI. And we have to, and we've made a promise to our patients who have enrolled in this study and been consented in this study that the data that we'll make available is de-identified from them. And so that's a significant challenge, is that when we collect data, we have to ensure that the data is also de-identified. It's a big initiative. We have efforts to recruit at a number of different clinical sites across the country. There's a central biorepository of biospecimens. We have funded what we call tissue interrogation sites, which are doing advanced, biotechnology on data, transcriptomics, single cell sequencing, proteomics, metabolomics, histopathology imaging, and other kinds of data like that. We then collect that data and integrate it with the clinical data, and that data then drives into both being made available to investigators, but also to populating a publicly available kidney tissue atlas, which has variables that are de-identified that come from that are sourced from these individuals. The data management kind of pipeline follows a pretty standard protocol, and actually this looks very much like the first slide I showed you about how we do data sharing. We generate data, and that data those data generators are aligned with the types of data that we're collecting. So we collect longitudinal clinical data with a research coordinator through REDCap. We have specimen tracking that is collected in a specimen tracking system. We have omics data, which gets uploaded with standardized metadata into a repository. The imaging data is uploaded and managed with standardized metadata, and electronic health record data is contributed in a standardized data model at each site that is aligned with the data model called OMOP, if you're familiar with use of EHR data. That data then is uploaded into a data lake, which can have raw data, kind of intermediate data sets that are been partially or fully analyzed, and then curated data. Where we're driving toward, we haven't actually operationalized this yet, is developing virtual sandboxes or enclaves where analysis on data can be done without data sharing outside of our environments. We're doing this we want to make analysis of PHI or potentially identifying information of these individuals as easy as possible for the investigator. And so by creating these cloud-like environments, we think we can do analysis on this data without having to worry too much about the data sharing issues, because the data never leaves our data lake environment. We have an honest broker, which sits between the potentially identifying information and the investigators, and the honest broker releases data into something that we call the knowledge environment, which is de-identified, fully QC'd, basically ready to use data that is integrated, patient level, and really is the product of the KPMP to be made available. And the knowledge environment feeds into repositories like the Kidney Tissue Atlas, which you can read more about, was just published in Nature a month ago or so. And I want to mention just a quick comment on these enclaves, because I think this is something that's new that we're doing. And I think the details of this workflow are probably too much to worry about here today. But what we're trying to do is make data sharing easier and more open for clinical data that has patient level information in it, even though it's been de-identified, or it has more like a kind of a HIPAA limited data set where we've removed the direct identifiers, but there may be indirect identifiers that are available. We're developing kind of an electronic click-through DUA process. It's kind of like you think about like clinical research is getting e-consents that are happening. We're also building e-DUAs, which enable, basically just like when you access like a new piece of software and they give you a EULA that you have to agree to that is like 20 pages of legal text that no one reads. We want to make this actually easy so people do read it, but we want to be able to enable clicking through to get a DUA between the investigator and the coordinating center, and then get access to integrated data in a cloud virtual environment that'll have access to all the tools that they normally use, like RStudio and others. All right, and so I think that's really important. We're still working on this, and there's a lot of challenges with it, but I think you kind of get the idea of like non-standard ways that we do data sharing to do analysis on data. I'm out of time, so I'm just going to briefly talk about the National Alzheimer's Coordinating Program. There are 3780 research centers nationally that collect clinical and phenotypic data from 80 patients. And we have 50,000 participants or so in our database that all have standardized data. It's very fair already because it's all collected in the same way. It's called the Uniform Data Set. And we've building and have largely built a cloud platform that is kind of our next generation cyber infrastructure platform for collecting data, making data available through a data front door that then can serve all of our constituents, everywhere from investigators to other coordinating centers to the centers themselves and the federal government. And that's ongoing. And I can talk more about that in the future if you'd like it. But this data has been used now for a number of machine learning, data science, and other applications within the Alzheimer's world to advance Alzheimer's research. I think I'm out of time. So I'm going to just finish up by saying there's this kind of challenge too that we see around real world data versus research data. Real world data is data that's not collected for the purposes of research, and we make it research data. And how do we integrate those together? How do we leverage them? I think is really, is an important problem. Okay. I'm not going to talk about UW Medicine. I'll finish up because I ran out of time, but I do want to thank everybody in my group and the ADRC and the NAC and the ADRCs that really help the research that we do. And I'd love to have a conversation about kind of our challenges that we see and any questions that you might have. And I really appreciate the invite to come and present.
Awesome. Thank you so much. I'm going to stop the recording now.